Thanks. Well, we're very excited to talk to you today about the next generation of approaches of regenerative medicine using induced pluripotent stem cells. We have a really great panel today. We're joined by Kevin Alessandri, who's the Chief Technology Officer of TreeFrog Therapeutics, Joe Brewer, the SVP of Allogeneic Research at Adaptimmune, Emile Nuwaysir, the President and CEO of BlueRock Therapeutics, and Alberto Santagostino, the SVP and Head of Cell and Gene Therapy at Lonza. I've asked each of the panelists to give a brief intro to themselves and to their technologies, and I'll start by asking Emile to kick it off. Thank you very much.
Excellent. Hello there. I'm Emile Nuwaysir. I'm the President and CEO of BlueRock Therapeutics. I guess I'd summarize briefly by saying that BlueRock started with the realization or the simple admission that degenerative disease isn't really curable, or even you really can't even intervene when you have a degenerative tissue, that we need a different approach to these diseases that we've labeled intractable, because all we can do is manage symptoms, diseases like heart failure or Parkinson's. We needed a new approach, and we think that the engineered cell, the approach that BlueRock is pioneering, is very promising in that regard to restore function and restore health. And the idea is that we know that the engineered cell by itself is this incredibly powerful new modality. I mean, we've captured one cell, the T cell, and one gene, the chimeric antigen receptor, under industrial control, and we've radically altered the landscape for oncology, for how we treat oncology. And the question we asked was, well, what could happen if we could capture, in theory, all cells and all genes under industrial control, if we could effectively manufacture any cell and edit any gene for any therapeutic purpose. Now, I know that's a rather broad statement, but that was the mission statement for the company. And we call this platform Cell Plus Gene. The idea is that we have reprogramming under industrial control with our own proprietary footprint-free reprogramming technology. And we have a broad and enabling license from Editas Medicine for CRISPR-Cas editing, and we're continuing to follow that field. And those two axes together, any cell and any gene, is the idea. Now, within that space, you obviously have to pick your battles, because there's lots of opportunity there. We're focused today in three areas: neurology, cardiology, and immunology. And in neurology, we're very excited to say we are approved to proceed with our clinical trial for Parkinson's, where we intend to restore the neurons that are lost, the midbrain dopaminergic neurons, and we hope restore motor control to the Parkinson's patient. We have three other programs in neurology. We also have a very advanced program in heart failure, where we intend to remuscularize the human heart by putting heart muscle cells back in. And finally, we have multiple programs in immunology, which we'd love to talk about too. So that's what we're doing.
Thanks, Emile. Joe, can I pass the microphone to you next?
Hey, so I'm Joe Brewer. I'm the SVP of Allogeneic Research at Adaptimmune. And Adaptimmune's really focused on making transformative cell therapies for cancer. So we use T cells, and we started out in the autologous space, where we're looking at taking patient T cells, re-engineering them so that they express a T cell receptor. So we work on TCRs rather than CARs at Adaptimmune. And then we send those cells back to the patients. So we've started looking at MAGE-A4 as an antigen, where we're using these therapies in synovial sarcoma, and we're really hoping to push that to registration as soon as we can. And these things they're changing people's lives, as Emile said, and it's a really exciting space to be in. And whilst we started out with that autologous therapy as working nicely in some patients, there are lots of issues with autologous that make it quite hard. It takes a lot of time from the patient's point of view. You have to get into hospital, have a leukapheresis, have your blood cells taken, and you send those off for manufacturing. And sometimes we can't manufacture from every patient. I mean, the patients have been through an awful lot by the time they come onto cell therapy. They've undergone lots of different chemo, they're different ages, their disease. You know, it has a real effect on their immune system. And that's an issue when you're using that as your starting material to make their products. So I don't want to underestimate how far the field has come, and there's been some absolutely amazing products out there. But ultimately, that all takes a lot of time, and some of these patients, they don't have that time, especially in the solid tumor space, where, you know, by the time patients come onto trial, they really are, you know, at death's door, some of them, and they can't afford to wait for us to do that manufacturing. So it's a really important part of Adaptimmune strategy is to get an allogeneic product off the ground. So we want to be able to give people cells on demand. So when a patient comes in and they screen positive for something that one of our TCRs could help them with, we want to be able to send them their cells like the next day, so that they don't have to wait. So to do that, you need an allogeneic therapy. You need to come off the shelf. You need to take the patient out of the manufacturing space. And the other great thing about doing that is not that it's a lot less invasive for the patient and it's faster for the patient, but it also allows you to bring in some consistency. So you can screen what material you start with, and you're hoping to get something where you make a single batch that will treat multiple patients, which is great, because then you are looking at driving down cost of goods as well. But I think really the most important thing is the consistency, and it's that ability to try and make every batch the same. Whereas at the moment, we're in a space where every batch is completely different. It's made different starting material every single time. And I think stem cells are a real key part of this future. It's a really exciting space to be in because they offer you that really broad flexibility to do lots and lots of editing. You can make lots of modifications, and you can really learn from what's working well in patients, what you need to improve. You can go back and do that in a stem cell where you control the donor. So you can work with one single donor, create a whole platform for multiple products that come forward. And it's an iterative building process, which again is a really good way of learning from the patient experience, learning from clinical trials and feeding that back into research. So I think there's an awful lot of power in this part of the therapy, and it's an exciting place to be. Thank you.
Thanks, Joe. Kevin?
Yeah, so I'm Kevin Alessandri, and I'm the CTO of TreeFrog Therapeutic. So we are a very young French company, as you could have guessed with my accent. And first, I want to thank you to be part of this panel because I think you guys are actually making it happen, and I measure my word. So it's really cool to be there today. And with TreeFrog, basically our motto is cell therapy for all. So I could not agree more with Joe. And our aim is TreeFrog is how we're going to manufacture cell therapy and to deliver them not to few hundreds of patients, but really to literally millions of patients. And that's quite an industrial challenge. And basically, we have a bit of twist in the story is that, in fact, I'm a physicist and my co-founder is a stem cell biologist, Maxime Feyeux. He's in stem cell biology since 18 years. In fact, he was the first Ph.D. in stem cell therapy in France. And we worked together since almost a decade. And basically, we came to the very fact that to make it happen, the issue is that for now, we grow cells directly in the machines, either in 2D, either in spheroids. Cells are directly exposed during the process. And we came to the point that basically that's where is coming most of the variability and the scale issue. And what we developed is very trivial. It's called system. C for capsule and stem for stem cells. So it's based on the encapsulation of cells. And basically what we are doing is that we are reproducing a biomimetic environment within the capsule. And this biomimetic environment, it makes a better yield of production of cell, better quality. For example, with stem cells, it will maintain the pluripotency, the genomic integrity. And the second benefit is that the capsule will protect the cells during the process of production. Basically, the media can go through the shell, but the shear stress will be only on the shell. And this will protect the cells throughout all the process. And thus, we have basically no more scale issues. And so it means that we can make much bigger batch with the highest quality possible. And that's where we think is the next generation for manufacturing in cell therapy. And we are a biotech. We have, so to push this technology, we have a pipeline of products. And basically, our lead program is in Parkinson's cell therapy, where we graft mature neurons microtissues. We have also a program, obviously, in cardiomyocyte, because obviously in cardiomyocyte, the yield and the scale-up is of utmost importance. And we have also a program in hematopoietic stem cells for grafting in bone marrow, where this time it's more the biomimetic environment which is required to reproduce functional hematopoietic stem cells for the grafting. Thank you very much for the invitation.
Thank you, Kevin. Alberto?
Thank you much, Gregory. It's very nice to be part of this panel with the other colleagues. And I am the Senior Vice President of Cell and Gene Technologies business unit in Lonza. As you may know, we are a CDMO and we provide services of development and manufacturing for companies. And it's great to have the opportunity to add maybe a little bit more the perspective of CMC technical development to the discussion that is going to come. As a company, Lonza has been very much invested into the iPSCs, and we have an history that is about 10 years old in developing this type of therapeutics. In fact, one of the leading cell lines that is even driven forward by NIH has been developed in our labs. One of the key things is that we really believe in the investment into the R&D, into process R&D. We obviously don't work in any therapeutics because that's the task of our customer, but we are really very much focused into the development part of the CMC technical development. So what do is that we have developed processes in terms of starting with tissue acquisition. That is a very important part to ensure GMP compliance, especially because in this case, this one donation will be at the basis of all the products that the company will be making, opposite to the autologous, as much so into the development of the induced pluripotent stem cells so that we can create a bank. And then from that bank, and quite importantly, then what is going to be the real industrial process of differentiation. And each of these areas, it has been a focus areas of development that is very important for whoever wants to develop then a therapeutic keeps that in mind. Because as much Joe said and Emile said, I shared the vision about what's going to be next, because doing an autologous is quite a cumbersome process. You need to run around every single patient, collect material, and then delivering them back with all the complexity and the cost that we know, to a point that someone would argue that it's even difficult to fully characterize those products in terms of specificity, because there is very much variability in the starting material. Allogeneic is the next wave and is what many companies are working on. But even in this case, we have a sort of a limitation on the number of expansion that are possible, could be in the 100, maybe in the thousands. And so even in this case, an iterative process of reproducing the starting material and then providing it to patients, so it doesn't solve all the problem. The holy grail will eventually be in iPSCs and in a situation where we can have a model very similar to the existing pharmaceutical, where we can have fundamentally off-the-shelf treatment or almost like that produced at the industrial scale. And as said before, there are the 3 steps: a very careful sourcing of the material, what we call tissue acquisition. There is a second step that is the creation of a bank, which is going to be eventually what you're going to use going forward as a starting material. And the third step is the differentiation process. And then I suppose we are going to get into the details, but the technologies like in bioreactor differentiation or the development of dedicated, fully owned NIT clean cell lines are particularly important for then the long-term success of the companies.
Thanks, Alberto. Before we head on to the next section, I do realize I was supposed to introduce myself, but I didn't. For those of you I haven't met, I'm Gregory Block, the SVP of Corporate Development at Notch Therapeutics. We are also an allogeneic cell therapy company based out of Vancouver, British Columbia in Canada, with offices in Toronto and Seattle. And our jam is T cells. So, you know, that's where our core focus is. So with that, I will delve into our first set of questions. And in this first section, I thought we would talk about, you know, we often talk about iPSCs as being the holy grail because they can be, you know, grown indefinitely and differentiated at large scale. But that comes with constraints. So the first set of questions are really to understand these constraints in iPSC manufacturing and the technologies that are emerging that will help us scale and differentiate to meet demand. So my first question I'm going to direct to Kevin. And you know, maybe you could talk a little bit about how TreeFrog is positioning themselves to sort of help companies and academics produce these cells at scale. And you know, with that, some of the fundamental considerations that partners are teaching you and how you're adapting to this growing field, and sort of how you're leveraging that to build out your internal programs and what your considerations are.
Yeah, so thank you very much. So that's a question up to the point. And as I will do a bit as Alberto and focus on the technicities of that. So basically, we had a lot of interaction with academics, biotechs, pharmas, and it's quite a diverse field, I should start with, because it's not just the variability is not just on the iPS cells, the starting material. And what we look so far is that the first step is exactly as you said, Alberto, the banking. So the first point we see, which is very common, is how to make a large-scale banking. The second point for iPS, really to reach mass market. And the second point we saw was, is there's an ability to have a scale-up technology, but which is compatible both with what I would call 2D protocols, but basically the ones that were developed on Petri dish, and the ones that are more in 3D, like spheroid protocol, which is often the case in cardiomyocyte, for example. And then the third point is that very often we are asked about the way we could control the product, meaning are we able to make fully mature cells and to ensure that they are delivered as such in the body of the patient? Because in the end of the day, the treatment has to be the cells, the functional cells incorporated in the body of the patient. And that's the three challenges that we try to address with system again. So this technology, as said Alberto, I developed this technology with a clear aim to be compatible with GMP standard of pharmaceutical industry, which means also the way how it works. So it's automated, closed, single use, and it's more on the side of the technology. Then in terms of pipeline, it means that we will aim the cell therapy, which need a lot of cells, like cardiomyocyte or liver indication, because that's where really there's a gap for now with cell therapy, because basically it's very hard first to make a bank of iPS, because as you said, the number of doubling is limited. And basically, you have to make the best out of yourself your cells, in a limited number of doubling. And so far, we managed to reach up to a doubling time, which is at 19 hours. So it makes like a factor of amplification of on about 200 per week with iPS, which is rather good. And then you have to be able to actually differentiate the cells in a bioreactor directly. And that at very large scale. And that's basically what we are doing now in the company. And because thanks to the protection of the capsule, basically we can make directly amplification followed by the differentiation within a single bioreactor with an automated process where you just have a sequence of media. And then finally, it's really this ability to bring those cells to millions of patients and to be able to deliver them. And we have this micro tissue strategy for grafting, which enables us to graft mature cells, basically. And now we think that our technology system, it will be part of the first technologies that will really enable the model of cell therapy, but as a mass market, not only as a niche indication. And that's really what we aim.
Thank you, Kevin. I'm going to continue the conversation by asking Alberto, Lonza has, as you mentioned in your intro, had a long history of supporting iPSC manufacturing, making the first GMP cell line, the NIH cell line. But there have been improvements in technological advances over the past few years. And I'm wondering if you can comment on how you think these improvements, are going to improve and advance the field. And you're on mute.
Very happy to do so. So one of the things that I believe is extraordinarily important is the aspect of differentiation and characterization, because on the differentiation aspect, there is an important impact that can actually be achieved in terms of the therapeutic side, on top of the safety side, because one of the biggest questions that is hanging out there is what is the consequence of having undifferentiated iPSC cells that are left into the mix after differentiation, which may be less of a problem in case of technologies that foresees encapsulation or anyway containment of the very same cells. Let's think about the production of cytokines like, I don't know, insulin production or even hemophilia factor production, where you can actually encapsulate the cells. But if you want to move into an actual broader application of real regenerative medicine, as Emile was mentioning, we need to feel comfortable to apply these cells in the body without any particular restriction. So there are many researches and activities that we are doing into both pushing the differentiation to the extreme so that we have almost no leftover or virtually non-detectable leftovers of undifferentiated iPSCs, but as much also on the aspect of characterization, which is always important, but is going to become extraordinarily important in the journey to GMP. So we have developed ever-improving protocols, both of characterizing iPSCs as such, with all the markers that are relevant to make sure that they are healthy, growing, in good shape, and effective, as much as to monitor that then we are completely eliminating them after differentiation while preserving a well-characterized and very vital differentiated cell types. On top of these, in terms of characterization, it will become very important the detection assays to make sure that the level of undifferentiated iPSCs are below a certain grade. And then the other aspect that we have probably learned and developed quite a lot is in the sourcing of the materials and in the tissue acquisition. I'm sure that you are reading the news and the challenges that many companies in the allogenic fields are facing now from a regulatory standpoint, because banks that have been done many years ago are not anymore living up to the standards of FDA and regulators, which is, by the way, a proper challenge and push from the regulators, because one thing if you, with a certain tissue, are treating one patient, another thing is with a certain tissue you're treating 100 patients, a different thing is with the same tissue, you're going to treat virtually infinite number of patients. So an effort of really going into an effective tissue acquisition and bank creation is particularly important. And the legislation is very different from any country that you are going to go. So Japan, very different than US, very different than EMEA, with requirements that go from mad cows to many other aspects that are worth to keep in consideration. Beside that, then we can go into other lines of research, like in bioreactor growth, yield, titer, and effectiveness, like with an ever-growing scale that we are investing to manufacture those cells, so that every batch is potentially even larger than the before, with a consequential decrease in cost per unit. But let me stop here.
Thanks, Alberto. Next question is to Joe, and we'll talk more about sorry, I'm not reading my question properly because okay. So we've got, you guys are working on iPSC-derived T cells. And this is, of course, happening on the backdrop of a lot of effort being put into allogeneic technologies to turn donor-derived T cells into allogeneic products. And I'm wondering if you could talk a little bit about some of the hurdles that the iPSC field will have to take into account based on those ongoing trials.
Sure. So I think, as Alberto said, the FDA do look at these things quite differently, bearing in mind the number of patients that you could potentially treat with a single batch. But when it comes to iPSCs, you know, the field, we still haven't proven ourselves, certainly not in the T cell space. I think it's more advanced in other areas, but in the T cell space, it's more of an unknown. And we're looking at, well, what really is the right thing that you actually need? So even in the autologous space, we know that we have products that work better in some patients than others. What's the critical factor? And we're talking about what are your criteria for saying this is a good product versus a bad one. You know, even the autologous field, they haven't defined those parameters. Likewise, the healthy donor derived, what's the true parameter that you care most about? Is it looking at CD3 and TCR positive? That's a really basic, or, you know, CD3 and CAR positive. That's a very basic characterization. But there are theories out there about, you know, is it T stem cell memory, T central memory? The the story changes over time as people are finding out more and more things, and it's probably quite context dependent. So that's also in the context of the tumor that you're fighting, and the cells that you're getting from the patient. And if you take a step away into a healthy donor, you're like, okay, so you can control the age of your healthy donors. You can control maybe what diseases they've had, what vaccination programs they've been through, but still they're going to have a cold, or they're going to come up against a virus, or their immune systems will change over time. So even if you find a really great donor who looks brilliant one day, in 6 months or a year's time, you might not get the same results if you went back and took another leukapheresis batch from that donor. So it's not 100% consistent, which the stem cells offer that potential that we could get to that stage. But, as Alberto says, we've got to be really careful in how we create those banks and how we qualify those banks. And we really need to understand our differentiation processes as well. And they've all got to be super tightly controlled to get that consistency. But consistency is, in theory, possible, but we just have to show that we can get there. And so it really is, to me, a lot of it is about variability. And when it comes down to the stem cell platforms, we've got to prove we can create the right sort of T cell. But we don't really know exactly what the right sort of T cell is today. I think the autologous system doesn't tell us that, and the healthy donor derived doesn't either, because with the iPSC platform, well, maybe it doesn't look exactly like an adult T cell. Is that a bad thing? Maybe it's got a little bit more innate activity. Well, the innate immune system is pretty good at killing things, so that's not necessarily a problem, as long as you can prove it's safe. And so really what we have with the stem cell platform is the ability to fine-tune a phenotype in a way that we don't have with healthy donors. With a healthy donor, you get what you get, and there are a certain number of things that maybe you can control, but a lot of things that you can't. But with a stem cell platform, there is a chance that you can get that fine control. Then the flip side of that is understanding exactly where you're aiming. And that, I think we don't know yet. Stem cell derived T cells are just coming in. Fate have built their platform on NK. They're moving into T cells. There's plenty of people in the T cell space all wanting to get there. But we don't have the clinical proof and the clinical validation yet. We're just on the cusp of that. And it was, the next 5 years are going to be fantastically interesting in understanding actually what sort of biology do these different differentiation processes generate, because everybody's got their slight twist on things. We don't know which one's actually going to be the best for patients. There's a kind of a broad thinking of, well, how do we get them to the tumor? How do we make sure that they work? How do we get them to react to the right cells? How do we get them to engage with everything else in the body? And also, when it comes down to differentiation and manufacturing, actually, can we do some of that in the patient themselves? So rather than doing it in a bioreactor, is there a stage earlier that you could say, well, we'll let them develop in the patient, and we'll let them do some of those doubling times effectively as the medicine, rather than before we actually dose the patients? Those are all huge variables that we don't know as a field, and we're only going to get there by understanding clinical practice and testing these cells in patients. So it is, it's a case of, let's get this past the regulators in a way that's safe and acceptable, and then we can start working on the biology, and we'll be really informed by the patients that we treat and what happens in those patients.
Thank you, Joe. Emile, the next one's coming to you. Given that you guys are poised for an IND, which is really exciting for our entire field, I'm interested to know what kind of strategic investments you've made to advance the manufacturing platform at BlueRock to get to where you are.
Sure. Well, I think the decision of whether to insource or outsource, whether to partner or do it yourself, I think depends a lot. There is no single answer. It depends on the indication, it depends on the cell type, it depends on your company's objectives, how you're financed. There's a lot. So for us, and it also, there's this simple I keep a simple rubric in my head, which is allo and auto and small and large dose. Okay, so our manufacturing world in IPS has 4 totally different manufacturing frameworks. And you start by saying, which one of those 4 are we in? So our lead program is an allo small dose product. That's the Parkinson's program. I would call that small because it's, let's say, roughly 1 million cells per dose. That's different from our second program, which is heart failure, where you're at 1 billion cells per dose, right? Which is 1,000 times the manufacturing challenge, to keep it simple. So, so you might choose different solutions for that. But from BlueRock's point of view, we wanted to build, we felt that the process development and manufacturing was key to the platform itself. At this early stage in the industry, we wanted to not only be the authentic cell expert and the engineering expert, but also be process development and manufacturing. So we've insourced for our lead program. So for the Parkinson's program, we can easily meet the, from a scale point of view, we can easily meet the clinical demand and probably the commercial demand today with the current process that we've industrialized. We easily make thousands of doses from a unit operation. So, or from a run, from a single run. So, we've built a pilot GMP facility. We have that facility in Toronto. We can manufacture our clinical product, and we are actually right now doing that. We're doubling the capacity of that facility this year. But the point is, we built that early stage plant under our control. We're actually also building a gene. That's to make cell products. We're building a similar facility to make gene-related products. The guides, RNAs, proteins that you need to do editing under GMP. We've got a separate facility for that. Those are both pilot facilities. And then by virtue of being acquired by Bayer, which I didn't mention, we are a wholly owned subsidiary of Bayer. But as a function of that, we have both the expertise and the capital to plan out our longer future. So with the help of Bayer and all of their manufacturing and process development expertise, engineering expertise, facilities, we're building a commercial facility. So we've already broke ground on that. It's moving forward, and we'll be able to manufacture our own commercial products. So we've chosen to insource the whole thing, and we see that as continuing. But I think you have to ask with each product, what's the best solution.
Thanks, Emile.
Mm-hmm.
Before we go on to the next section, I thought I'd throw a question out there to the group. And maybe it's based on something Alberto said. When you are an iPS cell company, you're faced with this daunting choice of, how to source your actual iPSC. And you mentioned that the regulatory environment creates a bit of a moving target. And you have to invest a lot into a program to then be told maybe later on that something is missing. And I'm wondering, how you guys are involved in the community or the discussions that are happening to sort of harmonize those efforts to understand so that people can make calculated decisions at the beginning and know that 10 years down the road, they're not going to run into problems.
Well, there are organizations working on this, the Standards Coordinating Body, the Alliance for Regenerative Medicine. There are various groups, either academic, industrial, that are working on trying to help both the community and the regulators to understand and set standards. And I think we all need to participate in that because it's key. CGMP, it is C, right? Current good manufacturing practice, meaning that it's a moving standard. Your obligation is to do the best you can with the limit of science today. That's all we can all do. But you do need to do the best. So I think we all need to participate in things like the Standards Coordinating Body. And for example, the Alliance for Regenerative Medicine will be publishing some reference documents on CMC guidelines for, it's called A-Cell and A-Gene, that'll be coming out. We can all contribute to improving those. So it's doing your part.
Thanks, Emile.
Yeah, I completely agree. If I just chip in a little bit there, but also I think the regulators at the moment are also being quite flexible with us. Like Emile says, it's current practice, and they, the interactions that we've had with the FDA and others, they are waiting to see what we deliver. So as long as we deliver something that's safe, certainly in phase 1, where we're just proving that these things actually work, they're prepared to give us some slack. And obviously, hopefully we'll be successful and we'll get to phase 3 in commercial. But there again, the regulations get tighter and tighter the further you go. So it's obviously something you don't want to reinvest in, regenerating a line to get to commercial. You want to be able to take that first one forward. You don't want to have to go back and make it again, because I think our limited experience says iPSC lines are not equivalent. You can't just get the same data with another line. There's quite a lot of heterogeneity there. There's quite a lot of variation. So it's really important that we actually can commercialize the lines that we're working on. And that is an ongoing conversation, I think. And, regulators will use those first clinical trials, that first data that we see in patients, and they'll react accordingly. So if it all proves that it's safe and it's fine, then maybe they won't bring in heavy regulation. But if it's not, we all have that responsibility to do things to the best of our ability to ensure patient safety, because this isn't a burgeoning field, and we could set it back for years if we do the wrong thing. I think then that's a responsibility all the companies in this space. It's on their shoulders to do the right thing.
Yeah, I completely agree with you, Joe. And so basically, there's also the GATE as organization, which is interesting for that. And so of course, we are in Europe, so I will speak about the EMA from what I know. But they are also very interested about the process-based more than the cell line-based, because basically, if we manage to have a reproducible way to make the cell line and to grow it with control of the risk, and especially, as you were saying, Alberto, a good way to characterize the cell line, that's in fact what they want. They don't want a therapy which is based on the cell line. That's just the way that they have to use, because it's what we have right now. But they want something characterized and in fact process-based, from what I understand.
Maybe just a couple of other add-on. This is one, if someone is seriously investing in iPSC, probably this is one of the parts where it's worth to overinvest in terms of creation of the line to minimize the chances of bad surprises on the other side, which means that there is a moving regulation, but there is probably also some judgment call that can be taken. There is I don't want to be tedious, but there is a panels of virus that could have an integrating effect against which might be worth to kind of test against at the very beginning and offset, to not discover that later on you find an integrated virus. There are considerations around potential diseases that might be considered not appropriate. So over-investing into the choice of where you take the line and how you test that line out is particularly important. As much so, the ability of characterizing the banks, which is particularly important, as Kevin said. It's worth to have a pretty broad panel of tests, because that is something that we want to be sure that is consistent. And then maybe the third comment that we have not yet elevated, but I still believe it's important, is the freedom to operate. Because you probably don't want to be in a situation that you're using the same iPSC cell line that is the same root in common to then a therapeutic that for whatever other reason got rejected, because then you will go all the way back to the same cell line, and then you might get into trouble without any wrongdoing. So some sort of freedom to operate and ownership of IP, which could be particularly expensive, need to be well considered in the strategies of the development. So I would say characterization, over-investing into the choice of the donor and testing out of the material, and then IP consideration should be probably the guiding star.
Thanks for your comments, everyone. I'm going to move to the next section of our discussion, which is a fun one, which is focused on, part of the Holy Grail and what Emile describes as a cell plus gene, which is designing your next generation products using gene editing. The first question will go to Adaptimmune. I think Adaptimmune is one of the companies I look at that has a really great vantage point, because you're running your autologous trials and you're in a position to learn. And so maybe you could comment a little bit about how you're leveraging that experience to design your next generation of products.
Sure. I think, that is one of our key advantages, and we use it every day. We are trying to learn as much as we can from our autologous patients to feed that into the Allogeneic platform, but also balanced with, until we've got that first line in the sand of how this Allogeneic therapy works, how these stem cell derived T cells work, actually we need to do a trial with those first so that we have comparative data. So there's a certain amount that you don't want to try and preempt, because we don't know how well these are going to work on their own before we add in the extra edits. But things like, we know that persistence, having the T cells for a long time really helps patients. And, you may be able to overcome that with repeat dosing, but if you lost the cells in the first place because they stopped seeing whatever it is they need to see, you know, have you knocked down the target at one point, then a repeat dose isn't going to do you any favors. Or have your cells stop working, you know, so how stable is their phenotype? And that's something we won't know until we get into patients to understand whether the allogeneic cells have a similar stability of phenotype as we see in our autologous or not. It might be slightly different. Certainly, people in the NK cell field are dealing on a very much shorter term basis, whereas T cells we know should live for years and years in patients ideally. And we see that in some patients in our autologous system, but not in all of them. So some patients lose their cells, and exactly why that is, you know, we don't know in every case. So persistence is a key one, but then there's also, we think about our next gens in allogeneic in the same way as we do in autologous. You know, there's getting to the tumor, you've given them a receptor to let them see the tumor first off, but you know, what else is the tumor throwing out? Is it about the inhibitory microenvironment? And there are plenty of ways that you can think of modifying your cells to overcome some of those things like TGF-beta, things like, you know, the adenosine inhibitory signals. Can you find ways of making your cells then overcome those? Making sure the cells get to the right place. You know, are they trafficking to the right place, or do they need help in trafficking? Are there things that you can do? And then one of the really big ones for me is actually bringing in other systems, other immune cells. You know, can you bring in innate immunity? Do you need it? Actually, is your phenotype a little bit more innate? Is that a good thing? But one of the real things then is, if you want to bring in the patient's own immune system into the equation, it's still got to be there. So that's overcoming lymphodepletion. At the moment, in all our T cell trials, we are really knocking down the patient's immune system before we give them the T cells to get those cells engrafting. If we can get them to engraft well without knocking out the patient's immune system, then that's really, it gives us a chance to use their dendritic cells, use their, you know, maybe they have tumor infiltrating lymphocytes. Maybe we can actually ramp those back up. Maybe there are ways that we can affect their, you know, their innate immunity as well. So I think there's an awful lot of things that are going on in tumors that it would be good for us to be able to, if you can get your cell there, you can get it to kick off an inflammatory cascade, but always you've got to balance that safety. You know, if you kick off a truly big inflammatory cascade, we're going into anaphylaxis, and clearly nobody wants to go there. So they're two sides of the same coin. It's not, you know, boosting your efficacy and making them work really well versus actually making them toxic to the patients, because we all know the immune system is a really powerful thing if it's not well controlled. So finding that fine line, that fine control, is really where these products need to be. And there are so many different biological pathways that you could be using. I think, you know, the advances in gene editing and the way we can modify ourselves have certainly one arm, but also our ability to do single cell analysis and to truly understand what's going on a cell by cell basis is really critical. But so much of it depends on actually getting snapshots of what's going on in the tumor and getting access to your cells in the right context. And I think one of the great advances in the past few years is that clinicians really understand the value of this now, and they understand how much information can be got from those sorts of samples, and they're so precious. But it really is getting the right sample and seeing your cells in action that tells you, you know, what's the next thing that you want to do with them.
Thank you, Joe. I'm going to direct the next question at Emile. You know, if BlueRock doesn't have that precedent of being able to run autologous trials, and so you're in a different position where you have to, you know, prioritize these targets and things you could do with your immense platform differently. Maybe you can talk about how you prioritize the seemingly endless things you could be doing.
Yeah, right. I think we think of this as actually 3 axes, not 2. I started out by saying there's the cell and the gene, but the reality is that that's the 2-dimensional version of the space we operate in. There's a 3rd axis, which is the systems biology. That is the nodes that control cell behavior. I mean, a cell is basically a super complicated, you can think of it as a super complicated genetic medicine, a gene delivery device, right? Except that it's integrating thousands of inputs to produce an output. It's way beyond our, you know, we can't engineer a cell in reverse. Like if you can't make one from scratch. So, but they're doing this by integrating all these sensory inputs and producing, for example, in our lead program, physiologic dopamine. So our approach to this is that we're going to try to step into this complexity with first the authentic cell. So just the pure floor plate derived midbrain dopaminergic neuron. We're going to put it back in the right location, restore physiologic dopamine. Okay. The second generation of that is that cell, but engineered for safety and persistence. So in that case, we see what we would call simple edits, like a kill switch, which is immensely complicated, actually. A kill switch or HLA editing for durability, which we see as a way to expand the applicability of the medicine and so make it so more patients can receive it. So if you think of the authentic cell or the engineered cell, but there's actually a step beyond that, which is the engineered cell where you've enhanced its function, it's delivering payload, it's sensing the microenvironment and responding dynamically. So it's only triggered in the right microenvironment with the right sequential signals. So you could make even safer or more potent medicine. So we're not jumping right to number 3. We're starting with number 1, going to number 2 and number 3. And then, you know, and then from there, you have to still simplify further because you even still just with an authentic cell have an infinite world effectively, right? So we think a lot about the cell disease link and do you really understand? It's sort of like target biology. Do you really understand the target in classic drug development? And do you understand the cell and this disease? Are they linked like a midbrain dopaminergic neuron? Can you make it? Then there's all these things about unmet need and for CMC issues, regulatory, all the things you would prioritize. We've focused on a strategy where, and once you've made that, now the real hard work comes in, which is how do you step into it? So we focus on a strategy where we early entry with a breakthrough product, followed by a best-in-class version of that product. And we think that that allows us to focus on these issues in a stepwise manner and to isolate the risks associated with each of those and focus on them as we mature and the industry matures. So for example, our lead product is an unengineered authentic midbrain dopaminergic neuron. The follow-on product is not. It's much more. So, so I mean, you could go, but you can, you know, you have to turn an infinite analog world of choices literally into a very digital thing where you're just going to do a stepwise approach. That's our approach. That's how we handle it. I don't know if that's what you're asking, but that's how we think about it. So.
Yeah, that's what I was asking.
Okay, good, because I got lost in my own answer there for the last, sorry, but it was fun. It's a fun question.
It's beautiful to have a vision, Emile, so why not?
Well, it's fun. It's good.
I've drawn all your axes on my notes.
Yeah, people make fun of me for doing that all the time. Never mind.
So my next question is directed to Alberto. You know, what from a manufacturing perspective, what unique challenges do you view existing for gene edited stem cell products? And what are you doing at Lonza to help sort of make that easier?
That's a very important question, but I'm gonna get to that, but I want to be inspired by what Emile said, if you allow me. So I think that what we'll be observing in iPSCs and in general in cell therapy is a journey that might be similar or not too different, or we can learn from the one of the proteins. No, because the very first proteins were substitution proteins. So I would expect that the very first iPSCs that we will see are just pure basic substitution of tissue that degenerated. And then we will kind of go into the complexity of adding functionality, and then we will go into the complexity of what probably we can call Emile system biology that we today observe in pathway engineering for the industrial biotechnology, like in the production of whatever products that we do in yeast or so on. But then that will kick in also in the iPSC. So I very much expect that the very first wave, and where probably it's worth to concentrate most of our effort today, is going to be in pure substitution. That is going to be of two types. The first type is cytokine production, and where you can technically encapsulate those cells so that you can get away a good share of minds of headaches related to characterization and protection of the system. To then go to the next baby step that will be making the cells as the actual product that substitute the tissues. So that's what I really believe in terms of evolution. So it will be very important to figure out a way in the first case to preserve efficacy and healthiness of these cells to produce their cytokine in a almost in vivo, but kind of separated environment. So all the permeations, for instance, when you have membranes. And in this second case, kind of really making sure that you have only differentiated tissue without any residual activity of iPSC. If you go to the next level around gene modification, I'm a big believer of characterization. So probably there will be probably some elements of full genome sequencing, or making sure that you are really working with single clones. So that is going to go into two ways: either really being sure about how you do your clones and your GMP manufacturing, and the other way into real characterization of the modification. Also because we know what we know and we don't know what we don't know, because you can have system genetic effects based on where you integrate, and maybe we just believe it's the same, but we just don't understand it. So going into the direction of really at least being able to map what we have done, it's a starting point to understand the consequence of what that will be happening. And then in terms of then how you will actually perform those things in manufacturing, it will always require a very proper sequential step in which you do things, so that you can always put a certain anchor on the quality of the previous step and that you are creating. So that would be more my approach to it in a certain manner.
Thanks, Alberto. Kevin, I'd like to ask you the next question, which is, what are ways that TreeFrog is envisioning using gene editing for some of your products? And more of a, a curiosity question of my own is like, do these, allogeneic packages, allow for you to gene edit the product? So, are there any technical limitations?
Yeah, so thank you very much for the question. So I will be a bit the bad guy in this section, so I will not exactly answer to your question. So even though we are a great fan of gene editing, and especially as you said, Emile, the system biology as a physicist, I'm very sensitive to that. But in fact, with my co-founder, we thought a lot about gene editing. In fact, he was talking with Laurence Dahéron recently, which is head of Harvard Stem Cell Institute iPS platform. And we have, he makes this kind of YouTube session where he talks with KOL. So you can see that on YouTube or on our website. And basically, he asked this very question to Laurence Dahéron, and she said exactly, we know already that there is a risk of genetic instability in iPSCs by the way they are generated, expanded, or during the differentiation. So manufacturing a safe cell is already extremely challenging. So now, if you add an extra step of gene editing, well, the risk is of getting additional genetic abnormalities is getting higher. So I completely agree with her, and Max as well. So basically, I think the base of gene editing is, are we able to generate self and reproducible iPS cells? That should be the base of everything. Because as is saying my COO in the company, Michael Lanero, he always saying, you don't make a racing horse out of a donkey. And so we cannot rescue the phenotype of an iPS. So I think that's the major challenge in gene editing. For example, I don't know, we all think about the super cell line, the one that would remove the immune suppression. But for this one, if we have such a cell line, we should be able to amplify it massively for all the markets, plus the creation. So there you have to be top notch in the way you grow the cells, like none we've seen so far. So I think that in terms of the field, as the industry, yes, gene editing is critical. It's most probably, as you said, Emile, I like very much the layering of technology, and I completely agree with you with this way to have the different version which will roll out. But we should focus on first solving this manufacturing issue around the quality of iPS. And in fact, it's feasible because in vivo iPS are clean. So there's a way to do it. It's not out of the blue. It's feasible. And I think that's a key point for gene editing, if you ask me.
Yeah, thanks, Kevin.
Sorry for the
No, I think I'm really glad you bring that up, because I think that's something that's on all of our minds in the field.
Yeah.
Is, not only how do we safely protect the genetic integrity of our cell line, because, DNA polymerase is your #1 enemy in that regard. But if we introduce these extra changes, what could happen, and how do we quality control that from a process standpoint is a major challenge we all have to be thinking about. I am keeping my eyes on the clock somewhat. And we have about 9 minutes left. And I do want to talk a little bit about the next section, but I'm going to focus down the conversation from the questions I had previously sent. The next questions are really set around the product development and patient experiences, right? We're now getting into a realm where these therapies are not just sci-fi technologies that we describe on the back of a napkin or we write in crazy novels. These are things that are entering the clinic. And we have to keep in mind our end user. And so I'm going to focus these next questions, one on Joe and one on Emile. And Joe, for the first one, I'm thinking more about the product development cycle. So, if you look at some of the early the early technologies for taking T cells into the clinic, these are using feeder cell lines. They're kind of complex. And some are using these allogeneic donor-derived cells, as we discussed previously. And I'm wondering, from your perspective, how you're positioning that, and also what you think the feedback from the regulators is going to be with some of these technological constraints.
Yeah, so for us, we wanted a process without a feeder cell line just because of the complexity that brings in. And it comes down to actually your characterization and your release, and how do you get it out, and how do you make sure you're only giving the right cells. But, to be honest, the regulators, again, because we're dealing with late stage cancer patients who really don't have any hope, regulators, when it comes to these particular class of patients, yes, first do no harm, but when somebody is at that stage of life where they're really desperate for something and they'll try a lot of different things, the question, it really is a balance of, risk benefit for that individual patient. And, so the benefit, if you can hold out something that really could help them, the risks have to go in line with that. So in cancer patients, that risk benefit equation is very different than it is, say, in a diabetic patient who could potentially live for, decades with insulin support treatment, or, but if somebody who's at late stage Parkinson's, again, they've already lost that function. So what do they have to lose versus what do they have to gain? And so that equation is different in different indications. And certainly the FDA and others seem to be open to that argument. It's a risk-based argument. It's an assessment of your product. Is it safe or not? And so, when it comes to T cells, giving an unaltered T cell is completely unethical, because if iPSC derived, that could see anything, and you're going to set off a massive graft versus host reaction, and that will most likely kill somebody. So there's no way we could take T cells in an unmodified state, and the regulators know that. So you have to modify it to remove the endogenous TCR. You have to modify it to make it safe. So we're already on that modification path. And it does seem to be how well you can characterize. It does come down to the quality of your characterization and your assessment of the risk. And, our Adaptimmune's history with regulators, even on the autologous, when we first started talking about the TCRs that we use, we went to them and said, we can't do an animal model. This is a human TCR, it only sees HLA. There's no animal model in the world we can run that would give us any safety. And we made that argument with them, and we went in with a fully in vitro preclinical package. And we've done that with our TCRs for a long time, because there's no sensible animal model that you can use. And the regulators were very open to that. And I've always found that regulators, to sound scientific argument backed up by data, then they're always open to that conversation. And so when we think, about iPSC derived, we're going with that same approach. We want to make the following edits for the following reason. And this is what we think. Likewise, as Alberto pointed out, having iPSC still present in the product would be of a concern, specifically because we are editing to make them hidden from the immune system, because we want them to last. So it comes down to characterization of our process is this long. We lose the iPSC signal at this point. We can no longer detect the cells here. this is, you know, and we continue the culture for longer. And so this is why we think it's safe. Do you agree? Those are the questions that we ask the FDA. But it's very product specific. So there's no one answer that they'll give that covers loads of people. You have to go with your own, this is our particular niche where our product is going to sit in this particular set of patients, and this is the safety data that we have. And it's a case-by-case assessment.
Thanks, Joe. You know, the last question I'm going to direct at Emile, and it was actually born out of a conversation we had in the lobby at the meeting on the med, which we can't go to this year. So I was thinking about it, and it was a meaningful conversation to me.
Was that the lobby bar, or that, because I'm just, go ahead.
I don't know. It's all a blur. It all smelled like cologne. That's all I remember.
Right.
So, you know, you guys are rapidly going towards the clinic for the DOPA trials. And, you know, there's an element here where you're dealing with the patient community and the patient population, both from your messaging as a company. You're now offering hope. I imagine that's been a really wonderful experience for you, for the company, for your employees, for patients. And I'm wondering if you could tell us a bit about your experience and what this field is going to be meaning to patients in the future.
Well, thank you for the chance to talk about that. I would just say, we take this, you know, we recognize that we're one of the first to try to stably engraft a manufactured cell, right? And that this is sort of a new era. And that we're, you know, we take that responsibility very seriously. I personally take that very seriously. And I think everybody at BlueRock understands how big a moment this is for us as a company, but there's 8 million Parkinson's patients where all we do is manage their symptoms. These are real people we know, family members, loved ones. I've seen it up close. I want to do something about it. So, you know, we're in the early stages of our trial. We haven't started actively engaging the community. It's just starting to ramp up. But I think our experience is that patients understand the concept of the cell is lost and that we could, you know, have the opportunity to replace it. They understand that at a basic level. There's a lot of noise about stem cells and the mischaracterization of that word. And so I've spent the last more than a decade of my life trying to re-educate the community, you know, investors, partners, everyone, and now patients on what that means, what that word stem cell means. So I think there's a little bit of education there, but patients in a visceral level understand what it means, the possibility to replace a cell and restore function. We've had tremendous response, not only from the patient community, but from the disease foundations. We have really good relationships with the foundations and research organizations. And we, you know, we've had a large number of people that have come to us, reached out to us, and it's very likely that some of those people will end up being included in our trial. So it's been it's an exciting time. We take it seriously. But we're really looking forward to it. There's been great support for the idea. We will not be the people that overpromise, and we will not be the ones that underdeliver. So this is about real people and a real disease, and you know, we're in it for the long term to make a difference. And I mean that for me and for everybody at BlueRock, and I'm sure for all of us. So super exciting. I don't want to take us down so much. Super exciting time for all of us. And the community's ready for these kinds of therapies if you talk about them correctly. So anyway, thanks for the chance to talk about that, because it's important.
Yeah, thanks for bringing us to the end of the session. That's the note I wanted to end on. This is an exciting time for everyone involved in this field. And I can't thank you enough for all of your time and for participating, and to the organizers for helping get us all together across the globe in a virtual setting in a strange time. And with that, I'll close out. I'll pass it to the moderator, or I am the moderator, but these other moderators on top of me to tell us what to do.
Thank you. Thanks, everybody.
Yeah, thank you.
Thank you very much.
Thank you.
Bye-bye.